All right. Hello, welcome to VSPG. Today we're having a presentation from Silvina Slagter at Yale University, doing her PhD, studying silica precipitation, and today is on experimental constraints on Ediacara-style preservation. Just a couple announcements before we really get moving. We are currently inviting people to give presentations after the new year for the winter and spring seasons. So if you have some people in mind, please go ahead and use the page on our website that is nominate a presenter, and just fill out that Google form for us, and we'll try to get to your speaker nominations. Yeah, it's really important for us to have the participation of you guys letting us know who you'd like to hear from. We also would really like to highlight the diversity in our research community as well, so keep that in mind when you're thinking about this, please. And yeah, the other thing Silvina would like us to mention is that for the Gordon Geobiology conference is coming up is for 2024, and it's going to be in Galveston, Texas this year. The abstract deadline is December 25th, or sorry, December 16th. Let's keep that one straight. Yeah, December 16th. So I was there last year when it was here in close to us in California on the coast, and it was just a fantastic time. I was there for the graduate student research part, but it was still fantastic. So recommend people sign up for that. Okay, with that, I'm just going to go ahead and introduce Silvina for us. So Silvina Slagter's research focuses on silica precipitation as a mechanism of fossil preservation, of fossil preservation, and as a chemical record of ancient ocean chemistry. One of her primary interests involves the taphonomy of Earth's earliest animals. She applies geochemical analysis in lab experiments and in investigation of Ediacaran fossils to better understand the nature of silica mineralization as part of the fossilization process. She also applies her research to study the evolution of marine silica cycling. Silvina completed her bachelor's degree in geology at the National University of La Plata in Argentina. For her master's degree, she studied silica sinter formation at the El Tacio geyser field, Chilean antiplano, using radiocarbon dating. And she is currently at Yale working on her PhD. Her thesis is titled An Experimental Approach to Reconstructing the Temporal Evolution of Marine Silica Cycling. Silvina, we're really glad to have you presenting today, and we're excited to hear about your work. So please go ahead and get us going with this.
Thank you so much. Thanks, Alex and Andrew, for the invitation, and thanks everybody for being here today. So probably you've heard before in this seminar series about the Ediacara Biota. The Ediacara Biota are really the first architecturally complex organisms that are preserved in the fossil record, and they were most likely soft-bodied organisms. You can see some images here on the left, and you can see that they had strange morphologies, and the affinities of these organisms have been extensively debated. And they have from millimeters to centimeters to sometimes 1 meter in size. And they are really important to understand the evolution and diversification of life on Earth, being an important stepping stone before the Cambrian radiation of life. So they are usually found in sandstone-dominated successions in the upper Ediacaran period around the globe. And although they are found in a variety of different ways of preservation, you can see some examples here. The most common way of preservation of these organisms is as Ediacara-style. And here you can see an example. Ediacara-style really refers to a three-dimensional casts and mold preservation, again, in sandstone-dominated successions. And because this is really unusual to find soft-bodied organisms in probably fine to medium grain sandstones, the style of preservation has been really debated. And people have proposed that these organisms experience rapid burial, and that's why they get very well preserved. But however, rapid burial is not unique for the Ediacaran. So there must have been another process that can explain the fine scale preservation of soft-bodied organisms in sandstones. And another point of view have proposed that these organisms may have had exceptional tissue consistency, meaning that they probably were more resistant to decay. But here I'm showing a variety of photos of this study from Evans 2019 that shows that, for example, Dickinsonia, which is one of the iconic fossils of the Ediacara Biota. Here you can see several examples of Dickinsonia preserved in sandstones. But these fossils are found twisted, torn, and unrolled, meaning that probably they didn't have any exceptional tissue consistency, and they rather were flexible and experience decay very fastly. So to understand how these organisms were preserved, also people have proposed that rather authigenic mineralization, that is the rapid precipitation of minerals surrounding these carcasses, was a mechanism that could explain fine detailed preservation of these fossils. Here you can see again Dickinsonia, a part in counterpart mold of these organisms. And based on the fossiliferous sandstones from the Ediacara Member in South Australia, some people have proposed that based on the presence of iron minerals surrounding these body fossils, the iron minerals were important for preserving these organisms. Here I'm showing a petrographic image of a fossil-bearing sandstones where you can see iron minerals surrounding grains of quartz. And this has also been known as the death mask model that proposed that veneers of pyrite precipitate onto the exterior of these body fossils and were later transformed to iron oxides. Other people have proposed that clay minerals were rather important for the preservation of these organisms, molding and binding the exterior of these body fossils. However, none of these minerals is present in all Ediacara localities, and also disentangling whether these are primary features or rather late-stage diagenetic features is really challenging. And that's why if we probably want to understand unusual distributions in time, we should rather look at unusual factors through time. And here I'm showing a diagram where you can see time in the x-axis and the dissolved marine silica concentrations in the y-axis, which is represented by this blue dashed line. So the dissolved marine concentrations in the ocean are thought to be very high during the Precambrian, and then after the radiation of silica-biomineralizers, such as sponges, radiolarians, and later diatoms, the dissolved silica concentrations drop down to really low values, such as in the modern ocean. And this has been suggested to be opening up a taphonomic window for the preservation of the Ediacara Biota when dissolved silica concentrations in the ocean were high. In addition, observations of paleontological, sedimentological, geological observations from the Ediacara Member in South Australia. Here you can see a bedding plane from the Ediacara Member, which is the type deposit for the Ediacara Biota, and also the richest in diversity for Ediacara fossils. When we look at these rocks, we can see here a plain view in A and a cross view of a fossil-bearing sandstone. And here the red dashed line is representing the area of an organism that is preserved in these quartzose sandstones. We can see that when we look at these rocks, the porosity is completely occluded by silica cements. And here, this is a cathodoluminescence image that essentially is showing that all these grains are quartz grains. There are some feldspars too, but essentially the porosity of these rocks is is completely occluded by silica cements. Therefore, it has been proposed that because the dissolved silica concentrations were high in the ocean, and we can see that these rocks are completely cemented by silica, the early silica cementation could have played a fundamental role in the preservation of the Ediacara Biota. And here I'm showing an outline of different tests that we run to test the model of early silica cementation, first using oxygen isotopes in the Ediacara Member, and then experimentally testing more closely this model that shows that early silica cements were important for the preservation. And then we test some roles of biofilms, substrates, and dissolved silica concentrations for this model. So first of all, we use oxygen isotope data to better understand the cementation history of the Ediacara Member. The cementation of the Ediacara Member is not really well constrained. The burial history of the Ediacara Member is not very well known, and oxygen isotopes can give us insights into the cementation process. And because of the monomineralic nature of these rocks, you can see here photomicrographs that are essentially showing that these rocks are completely formed by grains of quartz that are labeled here as DQ or detrital quartz, and OQ means overgrowth quartz. You can see that these are syntaxial cements that are cementing these sandstones, right? So we use oxygen isotopes, in situ oxygen isotopes, with colleagues of the University of Wisconsin at Madison to test this cementation model. But first, we want to take a better look at these rocks. Here I'm showing two cathodoluminescence images. First, I want you to take a look at A, where you can see that these grains of quartz labeled as DQ don't have grain-to-grain contacts and are rather embedded in a matrix of silica cements, which are labeled as OQ. And because of the limited grain-to-grain contacts and there are no pressure dissolution features, we can infer that these cements may have come from an external source that is not dissolution and reprecipitation of the grains. And the second thing that I want to highlight in B is that cements show a concentric zonation next to the grains. Here you can see a better close-up of how there are different stages of cementation that are clear because of the concentric zonation of overgrowth. So when we test the in situ measurements of oxygen isotopes in transects from grains through cements, there are a few things that I want to highlight. First, each of these points are in situ measurements of oxygen isotope, right? This is also a cathodoluminescence image that is showing euhedral cements next to the grains, and the colors of the colors of each grains are represented, of each point, sorry, is representing the value of oxygen isotope, which is labeled here in the inset. But essentially, the darker the color, the higher the delta 18O that we measure. Secondly, I'm here showing a histogram distribution of the overgrowth and the detrital quartz. And here you can see that the detrital quartz have a much lower value than the cements that have a much higher value compared to the grains, which is expected. But in addition, within the overgrowth, you can see that the overgrowth closer to the grains have a higher value compared to those farther away from the grains. And this is most likely representing a stage of cementation that closer to the grain is represented by lower temperature and then higher temperature through the cementation history. So another test that we run in these rocks, and this is not only for that image only, but also is represented throughout all the Ediacara Member and through different facies too. We also test for electron backscatter diffraction analysis to better take a look at the mineralogy here. In B and C, I'm showing cathodoluminescence images, again, that show a concentric zonation of overgrowth, right? And here I'm showing the oxygen isotopic composition throughout the cements and grains. And here, this color image is essentially an electron backscatter diffraction image that each color represents a orientation of the minerals with respect to a certain axis. And what is interesting is that the grains don't have the same orientations as the cements. And this is taking into consideration that all of this is quartz. I would expect to see that all the quartz have probably the same orientation. So the fact that it didn't, and this is also representative for many samples from the Ediacara Member, is probably telling us that the cements next to the grains precipitated probably as an amorphous phase that was later transformed to quartz through time. So what we observe with these oxygen isotopic values is that in transects from grains through cements, we see that the closer to the grains, we see a higher delta 18O value that represents a low temperature formation of the cements, and farther away from the grains, probably a later diagenetic stage of the cements. And this is important because if we know that these cements form at low temperature, we can probably infer that they had an important role in the preservation of the Ediacara fossils. Now, to experimentally test this early silica cementation model, we perform a variety of experiments in which we use soft-bodied organisms such as cyanobacteria, anemones, jellyfish, sponges, etc., that were exposed to high dissolved silica concentrations representing those inferred from the Ediacara, for the Ediacara seawater. And we held this experiment at constant pH and at constant temperature. And what we observe, here I'm showing results of two experiments, right? One of Phymanthus or an anemone and another one with a cyanobacteria through time. What we observe is the continuous precipitation of amorphous silica through time in the experiments. In the upper images, you can see that these images are from the experiments from time 0 to approximately 1 week. That is when these organisms decay. We observe visible silica precipitate, but also when we took a look at these samples under the microscope, we see that these experience precipitation of amorphous silica, first as silica nanospheres, and then they start to coalesce and form larger botroids of silica that later were visible with the naked eye. And to test whether these cements come from the seawater solution that we prepare, we could test that by looking at the chemical composition of our fluids. And here, essentially, what you're looking at is a graph that is showing the dissolved silica concentration in the experiment in the y-axis and time of the experiment in the x-axis. And each of these symbols represent a different organism that was what was used in the experiments. And what you can see is that the dissolved silica decreased through time at different extents into different organisms. And this was compared to a control experiment that didn't experience change through time, represented by the black diamonds here. And all of these happen in undersaturated conditions with respect to amorphous silica, especially if you look at the later parts of the experiment. Silica precipitation continued even in undersaturated conditions, which means that something was triggered the precipitation of amorphous silica. So to see what was happening in the experiments, we performed potentiometric acid-base titrations to have a test of how reactive the organic surfaces were to silica absorption. I'm showing here 6 different diagrams for the different specimens that were tested. And on the y-axis, you can see the site density, which is a measurement of the reactivity of the organic matter. these potentiometric acid-based titrations are essentially exposing these organic tissues to a protonation change, a pH change through time, and essentially see how these surfaces deprotonate. And we can model these results by looking at what functional groups are present in our samples. And we determined that for all of our samples, we had the presence of carboxyl functional groups, phosphoryl functional groups, and hydroxyl or amino groups. And you can see that in some of them, the bars are much higher than in others, meaning that some organisms have a higher reactivity to desorption of silica than others. But how does this relate to the silica precipitation, right? Well, here you can see the site density in the x-axis compared to the amount of silica that precipitated at the end of the experiments. And what you can see is that there is a positive correlation between the reactivity of the organic matter and the amount of silica precipitated, meaning that those specimens, for example, the anemone that we tested that have a higher surface reactivity experience the more silica precipitation. So this will mean that organics play an important role in the precipitation of amorphous silica and may have been responsible for this Ediacara-style preservation, forming the fine detailed casts and molds that we see in the Ediacara member, for example. But now these body fossils are usually associated with texture organic surfaces, or TOS, that are essentially the presence of microbial communities, or these are also known as microbial mats, or biofilms that carpet the Ediacaran seafloor. And here you see a variety of examples of body fossils associated with these texture organic surfaces. So to better understand what role these biofilms played in preservation, we also performed more experiments. And the presence of these microbial mats was important in the Precambrian. It was recognized early in the 90s by Slagter, that in this diagram, for example, show the garden of Ediacara, as they call it, where you can see that all these soft-bodied organisms that probably lived on the seafloor were associated with undermat miners, mat stickers, mat encrusters, mat scratchers. So a wide range of ecologies and behaviors associated with these microbial mats that later in the Cambrian decreased in abundance due to the appearance of bioturbation. So to test whether these microbes, as you can see here in a nice diagram, how all these green carpets were associated with different fossils or with different organisms of the Ediacara Biota that were later fossilized, we performed more experiments testing different microorganisms using two types of cyanobacteria and also different kinds of algae, also exposed to a high dissolved silica concentration resembling the Ediacara seawater at a constant pH and constant temperature. And for these experiments, we also use a quartz sand substrate. What we can see after approximately 1 week of the experiment is the precipitation of all this amorphous silica that is essentially represented here by this white smooth surface. And you can see how the filaments got entombed by silica. And interestingly, after these microbes decay, we could even see the formation of molds of these filaments, as is indicated by the arrows here. And furthermore, the areas of the experiments that were most populated by these microbes experienced more silica precipitation, that is in the lower right part of the image here. And when we combine not only microbes in these substrates, but also macroscopic animals such as these anemones, for example, we see that after approximately 1 week, we see the formation of molds. Here you can see the same experiments from a plane view and from a cross view, and you can see here the precipitation of silica on these organics, in these organic substrates. And even more so, after approximately 1 week, we see the formation of silica cements. This is an SEM image that is showing the precipitation of amorphous silica within the grains of quartz. So farther in the substrate, the silica triggered by the presence of organics is even filling up the pore space within the grains of quartz. So to better test whether these organics, such as we did for the potentiometric acid-base titrations, if the precipitation was associated with the presence of functional groups, we use Fourier transform infrared spectroscopy, or FTIR, in which we identify the presence of functional groups because they have a particular shape in the spectra of different organisms. Here, each spectra is corresponds to one of these organisms that we use for our experiments. And what you can see in the vertical dashed lines is the presence of hydroxyl groups here that has been previously identified, that the presence of hydroxyl groups associated with organic matter, here OM is organic matter, can have an important role for binding dissolved silica to the organic matter. Amino groups that were also identified through potentiometric acid-base titrations is present in the majority of the samples too, and can have an important effect on binding silica through electrostatic interactions. And finally, also the carboxyl groups that are associated with organic matter are present also in all the samples, and through cation bridging can also bind silica to the organic matter. So the fact that we see these filaments entomb really rapidly by silica, here I'm showing a scanning electron microscope image where you can see all these filaments entombed by amorphous silica, and the fact that we observe molds of filaments in Ediacaran-aged rocks can give us better insights in that these microbes were probably not only important for the ecology of these organisms, but also for the preservation of the Ediacara Biota. So to sum up this, the timing of silica precipitation was sufficiently rapid that outpasses degradation and can actually form a thin veneer of silica. Also, that microbial mats or biofilms that carpeted the seafloor were important lossy for silica precipitation, and the reactivity of the organic matter may explain the mechanisms by which these organisms were exceptionally well preserved. But now, moving into to the role of substrates and dissolved silica concentrations in the preservation, we have to highlight that not all Ediacara fossils are found in clean quartz sandstones, such as in the Ediacara Member in South Australia. There are also nice examples of Ediacara fossils preserved in finer grain sediments in more heterolytic successions, such as in this example here. And furthermore, it has been proposed that clay minerals could be associated with these fossils. Here I'm showing a image of LaPlante 2011, where you can see that these fossils, which are represented by this diagram here, have a characteristic chemical composition that may be associated with clay minerals. So we expand these experiments to try to better test what happened under different substrates, not only with quartz sands as we did before, but also using kaolinite and iron oxides. We use the same variety of organisms such as sponges, anemones, cyanobacteria. And because of the dissolved silica concentrations in the ocean in the Ediacaran are still not well constrained, you can see here in this recent paper that there are different interpretations for the dissolved silica concentrations in the ocean. Some of them propose a more stepwise decrease through time. Some others propose a more rather continuous decrease. And furthermore, there are variabilities between the estimations for the Ediacara dissolved seawater concentrations. So to have a better representation of how the different estimations for dissolved silica concentrations could play a role in the preservation, or what is really the threshold for the well-preserved organisms to happen. We also run experiments with a combination of these different substrates and 2, 1, and 0.5 millimolar dissolved silica concentrations. And what we observe, here I'm showing a variety of images from the experiments, is that in those experiments that we use quartz sand and iron oxides, we see the formation of amorphous silica precipitates that are indicated by these arrows associated with organics. But however, when we use kaolinite in our experiments and low dissolved silica concentrations, such as 0.5 and 1 millimolar, we see that kaolinite could rapidly cover the organic surfaces. Here you can see organics associated with platelets of kaolinite. And here this is a sponge spicule that is rapidly covered in platelets of kaolinite. But we also see the formation of amorphous silica in these experiments. However, when we use higher dissolved silica concentrations in primary kaolinite, we see the formation of what it looks to be an authigenic phase that is characterized by aluminum and silicon. So it's probably also a kaolinite, but in this case, authigenic, that is indicated by the arrows here, that can form rapidly in the experiments and is abundant throughout the experiments. But to have a better count of how much of these authigenic minerals is present in our experiments, what we did is at the end of the experiment, have a point counting of authigenic minerals. Here you can see a screenshot of the software that we are using that essentially randomly selects points throughout the images, and we assign whether those are clays or amorphous silica or what is the mineral that we observe. And in this case, for example, you can see that all the red points are kaolinite, primary kaolinite, and that the blue points are amorphous silica, and we calculate the relative abundance of those. In addition, what we do is try to compare what mineral we would expect to be precipitated out of our experimental solution using geochemical modeling. And in this case, trying to see what are the saturation indices for various minerals without any organics, and in this case, using 1 millimolar dissolved silica concentration, but we run this for all the different experiment setups that we have. And what we can see here is the saturation indices of various minerals. Here you can see the saturation index, right? That essentially this diagram shows that if the mineral is to the right of the zero, so essentially if it's positive, you would expect that mineral to precipitate. But if it's below zero, you wouldn't expect that mineral to precipitate, again, without any organics. So if you take a look at amorphous silica, for example, you can see that this mineral is undersaturated with respect to our experimental, in our experimental conditions. But however, we do observe this in our experiments. And if you may recall that the organics may have had an important role for precipitating amorphous silica, that may be the reason why we still observe amorphous silica precipitated. On the other hand, for example, kaolinite, it's a mineral that we would expect regardless of if there are organics or not in our experiments. So to sum up what we observed so far in experiments without organisms, there is no amorphous silica precipitation. But in experiments with quartz sand and iron oxides, we see amorphous silica precipitate with organic surfaces. And when we use low dissolved silica concentrations, we still see a good preservation of the organisms that we use. And amorphous silica is more abundant than for example, authigenic clays. But under high dissolved silica concentrations, these authigenic phases form in association with primary kaolinite, and they may have an important role for the preservation of these organics through time. So essentially what we observe, and here I'm summing up what we hypothesize for being the the preservation of these organisms is that at the beginning, these macroorganisms living in association with microbial mats, they could live under different substrate conditions. For example, here I'm showing 3 end members, one that is represented quartz sand, another that is represented quartz sand and primary kaolinite, and another of quartz and iron oxides. So depending on in which substrate they are associated with, you would expect different authigenic minerals to be important for the preservation. Here the organism is decaying, and essentially in A and C, which is quartz sand and quartz sand with iron oxides, we see amorphous silica precipitation, and in those cases that kaolinite is involved, we see an authigenic phase that is likely an aluminous silicate that could also have a potential important role for preservation. So in the end, you would have different substrates, right? Quartz and amorphous silica, quartz, iron oxides, and amorphous silica, and in B, quartz, kaolinite, and authigenic kaolinite associated with the formation of casts and molds as we see in Ediacara-style preservation. So to conclude, the global control on the Ediacara-style preservation may have been the high dissolved silica concentrations that, again, we don't truly know exactly what the concentrations were, but testing a wide range of dissolved silica concentrations, we think that the higher the dissolved silica concentrations, the better the preservation we observe in our experiments, and may have played a fundamental role in fossilization. Then important petrographic observations and also geochemical analysis like oxygen isotopes can support this early stage of cementation of the Ediacara biota. Then we also test the role of biofilms in Ediacara fossilization, and we found that they may have played a fundamental role in preservation, being important loci for the formation of amorphous silica. And finally, that under a wide range of substrates, we can expect this Ediacara-style preservation to be found in the rock record. So with that, I would like to thank you all very much for being here. I would like to thank my advisor, my collaborators, and of course, my funding sources, and all of you for being here, and I'm happy to take any questions.
All right, thanks, Silvina. That was really good. So we'll go ahead and switch to question discussion. Silvina, we don't usually stop that until all the questions are asked. So if you need to get going sooner, just let me know. So you can raise your hand with the button at the bottom if you find reactions, and click that. It should be the big raise hand button there, or you can type it into the chat and I'll ask it for you. But we have a first one from Greg Vitalik. Please go ahead, Greg.
Oh, that was great, Silvina. I enjoyed that very much. For the first time, I noticed you had oak leaves in the mix there, which fascinates me because we have literally millions of silicified oak leaves throughout the rock record of oaks, going back almost to the Cretaceous. And yet they didn't seem to take up as much silica from those selected sites as the jellyfish. We have no silicified jellyfish at all in the entire fossil record. Am I misreading those data?
No, so it's interesting that you bring that up because the oak leaf that we use in our experiments was kind of to have an end member of our recalcitrant organic matter that didn't degrade very rapidly, and that is probably why we didn't observe any silica precipitation on that. And on the other hand, the jellyfish or the anemone or the other soft-bodied organisms that we use in our experiments did get degraded more rapidly and experience more silica precipitation. However, yeah, does that answer your question? Or
not entirely, because I've always been under the Barghorn and Leo model that plant tissue especially really attracts silica, and you're showing just the opposite. which leads me to my next question. If organisms are involved, and there has been a really good case made by Birnbaum, among others, that sulfur-reducing bacteria are involved and that they actually precipitate spicules of silica that do the job. Do you have any idea what organisms are involved in this early precipitation?
So I'm familiarized, of course, with the Leo and Bajardon model, but do you mean in my experiments what organisms?
Yes. I mean, there are presumably bacteria that are doing this. I'm not so sure that the organisms that are your experimental subjects are actually doing the silicification because they're being silicified. You see what I mean? They're not sterile cultures, right?
Right, so the organisms as well as the microorganisms that we use in our experiment had functional groups that are reacted to dissolved silica, and probably that is why is essentially binding the dissolved silica to the organic matter. But I guess the microbes would have a better have a higher reactivity and therefore have a better preservation in that case. And we use a variety of different microorganisms, right? From cyanobacteria to algae, and all of them seems to have a important reactivity for dissolved silica, and therefore for
well, that does make sense because we do have a fantastic record of silicified cyanobacteria and others going way back in the Precambrian and even in the Phanerozoic. So, yeah, that's really fascinating. Thanks so much.
Okay. No hands up at the moment.
Well, if nobody is flipping up, maybe I can. I have several questions, and maybe you can help me. So silica level decreased in early Paleozoic, and wouldn't you expect that Ediacaran style of preservation at least extend into early Cambrian? And is there something consistent with it in the rock record? So that's maybe first question.
Yeah, so that's a very interesting question because we don't exactly know when the dissolved silica dropped, right? We estimate that the radiation of sponges and later radiolarians play a fundamental role, but exactly when and in what magnitude was that decrease, we exactly don't know. And we also see some examples of Ediacara-style preservation in the lower Paleozoic. There are some nice stranded medusae from the Cambrian in Wisconsin and also in New York. So probably the Ediacara style of preservation did get to the Cambrian.
Thanks. Like another question on oxygen isotopes. So you're trying to answer a question if it's early date, what role of seawater silica played in it, and I'm wondering if you would compare, like silica cement are very common in quartz sandstone, like of any age. And John Wally and his group did a lot of work on oxygen isotopes of cement. So I'm wondering if your highly positive values, are they different from values that you typically find in silica cement, like let's say in early Paleozoic, or it's the range of values is the same?
Yeah, no, that's a great question, and somebody that I didn't went in further detail, but actually the values that we observe for the overgrowth are a little lower than we observed that previously reported quartz arenites for the Cambrian, for example. In Wisconsin, there are some analyses that were done there, and the average, let's say, was closer to 28 per mil or 25. In our case, our average was approximately 19 per mil. So it's a little lower for the earliest, most proximal to the grain cements. And that is why we got thinking about whether we are representing, whether we should read those values like at face value and interpret those at the temperature of formation of quartz, or if they are rather representing a phase transition from a more amorphous phase to quartz later, in a later diagenetic stage. And that is why the EBSD data is very interesting, is because we see that they don't have the same crystallographic orientation with respect to the grains. And therefore, the values that we are recording in the most proximal to the grains may probably represent a transition from amorphous phases to a more crystalline phase later in the diagenetic history.
And another question, so maybe you can explain it a little bit. So at the end, you suggested that high dissolved silica concentrations, you would expect to have alumosilicates to be formed, but in Ediacaran style of preservation, you mostly saw silica cements. So how does it kind of work? Why, like in Ediacaran, when you expect to see high dissolved silica, you don't form this alumosilicates mineral that sort of consistent to your experiments? Maybe I missed something.
So let me see if I got the question. Probably what I'm trying to highlight here is that not all Ediacara localities have just clean quartz sandstone and just quartz overgrowth. There are some examples in which there are clays, and we are thinking that probably the formation of authigenic clays may also be responding to a high dissolved silica concentration in the ocean, right? And this may not only explain the mechanism by which the Ediacara member, which is pure quartz sandstone or a quartz ironite, but also in those other cases in which we find more finer grain sediments associated with the fossils of the Ediacara Biota.
Okay, so you basically mean if you have only clean quartz sandstone, you would form silica cement, but if it's sort of sandstone with some clay minerals, you would start forming alumosilicate minerals.
Yeah, exactly. And also you can form cements under in those experiments, for example, that we use quartz sand and kaolinite. We also see the formation of amorphous silica, but the question is to what extent one is, more abundant than the other. So I guess it would really depend on the substrate that you start with, whether you form either an authigenic clay or a more, quartz cemented sandstone. Yep.
Okay, thanks. There were a couple of questions in chat, so maybe I come back later.
Yeah, so we had one from, I'm sorry, Phil, if I mispronounced your last name, Phil Vixiboxy. Says, very interesting talk, Silvina. How much of a rheological effect do the silica gels have on sediment versus the rheological effect of EPS from biofilms on holding the grains together?
Yeah, no, that's a great question. I think, the EPS from biofilms, what it's doing is essentially being reactive to silica precipitation. So I guess the silica gels have a more important effect on holding the grains together. And microbial mats, when I presented the Slagter diagram that you had, very important microbial mats in the Ediacaran, and they decrease through time. They were proposed to have an important effect on binding the sediment. But I think that when the silica gel starts to precipitate, it's an important agent of cementation. So I think the gels will have a very important role in that case.
Okay, Greg, your hand was back up again. Did you want to ask another one?
Yes, I do. Yeah. I noticed that you have a slide in reserve there, which is one of my favorite ones. The next one along, with the germanium to silicon ratios.
Oh yeah, sorry.
Yeah. No, I love this slide. As you know, those levels of germanium to silicon in the cement are only found in soils and paleosols. So I have two questions. How do you explain that away if they're marine? And also, are you doing more of this? Because I'd love to see more of these data.
Well, I'm actually measuring the germanium to silicon ratios in the experiments to try to see if they actually the organophilic behavior of germanium is really capturing a high germanium to silicon ratio in those cements like closer to the grains compared to
Oh, fantastic. Is it working? Are you finding high germanium ratios in your experiments, in your seawater experiments?
Well, we still haven't finished them, so I'll be happy to, when I have results, to share them, of course. But we still don't have the results there yet. But your first question was referring to why do we see high germanium silica in the cement?
Right.
Yeah, so as here I'm showing the organophilic behavior of germanium. Of course, there is really little experimental evidence on how germanium works in these settings, and I would love to have better, like to have results on my germanium silicon in my experimental conditions. But really what this is showing, and this is another line of evidence that show that cements, as I show for the cathodoluminescence images, that there is a lot of cement in these rocks that unlimited grain-to-grain contact. So really the cement may have come from another source that is not dissolution and reprecipitation. And that is also what the germanium is showing here, that it has a very different signature with respect to the grains. So
yeah, I don't understand it either. I don't understand why soils have such high germanium content compared with all aquatic systems, but that's just an observation. And it turns out to be true on Mars as well. The paleosols on Mars have enormous germanium concentrations, and the sediment's low. I'd love to know why.
But yeah, no, I'm looking forward to have results and see really what's going on and how fast it can be taken up.
Fantastic, thanks.
Okay. Relevant to that, Phil also says, what is the germanium content of the feldspars? Could the germanium enriched cement not be derived from feldspar dissolution?
Oh, well, I don't have in the top of my head what is the germanium content if it was measured in situ for the feldspar, but I think these are mostly, quartz sandstones. So this trend is not only observed for like one area, it's observed for a lot of different transects from grain to cements. And I think the amount of feldspar probably can't account for the amount of germanium that we see in these transects. But that's an interesting question.
Okay, Andre, did you want to swing back around?
Yeah, just a comment. Did you or somebody else observe similar amorphous structures in early Paleozoic sandstones and overgrowths or in cement? And if it was observed, it would be very interesting to compare oxygen isotopes for this amorphous phase or sort of precursor amorphous phases to what you observed in your work.
Yeah, I haven't performed any oxygen isotope measurements in the experiments, but certainly it would be interesting to see if those can match what we observe for the cements in the rock record.
I meant not in experiments, but in early Paleozoic cements or overgrowths in sandstones. So like John Valley did, again, a lot of work on oxygen isotopes. So I'm wondering if they saw a similar indication for amorphous precursor as you see in your study. And when you can compare sort of the same type of textures in terms of oxygen isotopes.
Oh, so let me see if I got the question. If we observe the same isotopic values for Cambrian, for example, successions that in the Ediacaran. If you are asking if the textures are the same too?
Yes, so like did somebody observe that cement, so you explain difference in oxygen isotopes from Paleozoic by amorphous precursor. So was this amorphous precursor ever observed in early Paleozoic sandstones, and if so, do they have similar oxygen isotope values?
Oh, yeah, now I understood. Sorry, I didn't catch that properly in your first question. But in the Cambrian analysis or Cambrian Ordovician, or like early Paleozoic sandstones from, for example, Wisconsin, they didn't do any EBSD analysis as far as I'm aware. So they didn't observe any, different crystallinity between the grains and cements. So they didn't interpret any amorphous precursor, but this is really something that we don't know. That's for sure. Like the thing that they show in their papers is that quartz could be precipitated at much lower temperature than previously thought, but it's really, as I think, a still open question whether those observed for the Cambrian or Ordovician could have had a amorphous precursor. We don't know. Yeah, that's a great question.
And the last question following on Greg, but maybe you already answered it. So one of your diagrams showed that demosponge and I think medusoids were very efficient in removing silica from solution in your experiments. And I'm wondering if you can, from silica cements that you observe, infer anything about sort of biological affinity of Ediacaran fauna, but I guess it's very far-fetched and maybe like decomposition of organic matter enzymes and bacteria play a more important role.
Yeah, no, that's a great question. I think, we observe the amorphous silica precipitation for a wide range of different soft-bodied organisms. So I was also thinking if, it's more prone to be precipitated to one, let's say, one animal with respect to another. But we see some differences in, for example, the anemone have higher precipitation than, let's say, the sponge. But I don't think we can infer any, animal-like affinity or anything with respect to just looking at the amorphous silica, if that answers your question. But yeah, super interesting.
Thanks. Really interesting stuff.
So Daniel Segesman says, "Hi, Silvina, great talk. Very interesting and highly detailed work. I was wondering what your thoughts are on the possibility that the geochemical values you observed, such as high germanium, could be due to biotic effects that are adaptations or byproducts unique to the microorganisms making up the microbial mats in the Ediacaran."
Well, I think, that there is little experimental evidence of organics and germanium, but it has definitely been proposed that germanium has an organophilic behavior. So I think it could very well be associated with the microbes. Like microbes can take preferentially up germanium with respect to silicon, and that may explain the high germanium to silicon ratios in the cements. Whether it's only the microbial mats or the biofilms, or it's also the macroorganisms, we still don't know, but it's a great question. Yeah.
It kind of led to something I was thinking to ask that maybe I just missed. But so when you analyzed the cement in between grains, you mentioned it was fossiliferous layers. Was that in sand grains that were associated with a fossil?
Oh yeah, I should have probably clarified that. But yeah, these are all fossil-bearing sandstones in which we analyzed.
So you analyzed, so I guess my question would be, did you notice a difference between those that were associated with a fossil versus those not? I know there's basically mat on everything back in the Ediacaran, but so you have organic matter everywhere, but the question still stands that for me, I guess, is was there a difference between the fossil versus non-fossil, like a not fossil area without fossils?
In terms of oxygen isotope values, you mean?
Sure. Yep.
No, I mean, not really. We did this for different facies, for different fossiliferous beds, and we didn't observe any difference between the more closer to the fossil compared to farther away from the fossil.
Okay. Okay, that's cool. All right, thank you.
Yeah, no, thank you.
I don't see anything else nothing else was written and no hands up. So thanks a whole lot for the presentation today. It was really good and it pushed a lot of good discussion. So great to have you.
Thank you so much. And thank you everybody for being here.
All right, thanks everyone. Take care. We'll see you next week.